start? All right, cool. Should I start? Yeah. All right. Hi, everybody. Thanks for having me here. It's a real honor. My name is Omer Weisbrod. I'm a postdoc with Alkes Price at this Harvard School of Public Health. And today I will be speaking about using epigenetic data for GWAS interpretation. All right, so let's go. So I'm going to start kind of in a light way by first saying a few words about GWAS. So GWAS are genome-wide association studies, and we have lots of genome-wide association studies. So the idea is conceptually simple. We collect a large sample of individuals, and we genotype them, and we measure some phenotype or a bunch of phenotypes, and then we look for associations between these genotypes and the phenotypes. And here you can see the number of GWASs that we have between 2007 and 2017. So you can see that we have lots of GWASs, and the number keeps increasing from year to year. And you should pay special attention to these dark blue bars, which are the huge GWASs with over 100,000 people. So we get more and more of these GWASs year over year. So there are a lot of GWASs, there are lots of genetic data out there. And what do we learn from these GWASs? So one of the main things that we've learned so far is that, sorry, before saying that, I should also mention that not only do we have these GWASs, we also now are in the age of biobanks, and biobanks are these huge samples that are collected by countries or country-like entities. And the largest sample, largest biobank that we have to date is called the UK Biobank, which you may have heard about. So they collected genotyped and phenotyped around 500,000 people from the UK. So this is close to 1% of the UK population. And the data is relatively easy to access to researchers. So this is an amazing resource. So here you can see a map of the UK, and every dot here is the place of birth of one individual in this cohort. So we have lots and lots of genetic data. And so what do we learn from all this data? So one of the main things that we've learned is that most traits are really polygenic, meaning that they are affected by multiple variants, not just one. So when you read in the news that scientists found the, I don't know, the gene for height or for blood pressure or something, it's probably not true, because there is no one gene for anything. Everything is very polygenic. And to illustrate that, I'm going to show some results from a Twitter bot called GWASbot. So GWASbot is a Twitter bot that posts the results of one GWAS per day. You can follow it on Twitter. And just a few examples. So let's look at this example first. So GWASbot posts plots like this. So this is the results for a GWAS for blood pressure medication. And all right, so what can you see here? So on the x-axis, you can see genetic variants in the genome, or what I'm going to call SNPs from now on. So every dot like this is a SNP, and they're arranged according to their physical location on the genome in chromosomes, so chromosome 1, chromosome 2. And on the y-axis, you can see minus log 10 p-value of the statistical tests that ask whether some SNP is statistically associated with the phenotype. And you can see this red dashed line here. So everything above this red dashed line is statistically significant after multiple testing correction. And we impose a pretty stringent multiple testing correction because we test lots of different hypotheses. We assume that we have around 1 million effective SNPs, and so we correct for 1 million different statistical tests. So everything above the red line is statistically significant after multiple testing correction for 1 million hypotheses. And you can see that we have lots and lots and lots of SNPs that are statistically significant even after this very stringent multiple testing correction, right? And so this is not unique. This is what we typically see. So I'm going to show you a few more examples just to get you in the mood. Miserableness. So this is GWAS for the property of being miserable, according to a survey filled by the participants of the UK Biobank. You can see that we have lots of signal everywhere, and everything appears to have some genetic component, polygenic genetic component. So just two more light examples. Hot drink temperature is very, very polygenic. So some people like their temperature, their tea hot, and it's affected by genetics and cereal type. So basically everything. So this is just to get you in the mood. All right, so hopefully I convinced you that lots of stuff is genetic and polygenic. And the question is, what do we do with all these SNPs that we found? Because if we would have found, only one SNP in one gene, then we would simply try to understand what's going on in that gene, and that would be simple. But it's not as simple as that. So it turns out that there is a pretty limited role for protein coding regions in general in GWAS associations. So here are results that were published over 10 years ago already, but the main theme still stands, that protein coding regions play a limited role. So that was in 2009. There is something called the GWAS catalog. Back then it had only 465 GWAS associated SNPs. Now we already have probably tens of thousands. And back then they saw that only 11% of GWAS associated SNPs lie in coding regions. So coding regions account for 1% of the genome. So is that good or bad news? So on the one hand, you might consider it as bad news because it means that 89% of the GWAS associated SNPs are not in genes, are not in coding regions, and that makes interpretation much more difficult. We don't know what exactly they do. They probably regulate some other genes in some way, but it becomes complicated, right? So that's one thing. And the good news is that it's 11%, not 1%. So 1% means that these coding regions are completely irrelevant, right? So it means that the null hypothesis holds that there is no special role for these coding regions. So in fact, we have 11-fold enrichment. And if you dive deeper, they saw that 9% of these 11% were non-synonymous SNPs. So again, that confirms what we know about biology, that non-synonymous SNPs are important and they affect trait biology more than other SNPs. So it's both good and bad because we still have a lot of genetic signal coming from non-coding regions, and we don't really know how to interpret that. And so in order to help us interpret these results, we often use what we call functional annotations. So to understand what this means, here is a figure that I really like from a 2014 paper. And what you can see here is, all right, so you have this green blob here. So these are the set of SNPs that are actually affect some phenotype or the set of phenotypes that we care about. And let me pull the zoom window. All right, because now we can see. All right, cool. So, and we have multiple sources of evidence that could help us understand what these SNPs in the green blob are. So we have the set of protein coding SNPs or genes, so the set of SNPs in coding regions, which I already mentioned. And we have another source of evidence, this red circle, which are SNPs with evolutionary evidence. So for example, SNPs that are conserved in mammals, if they're conserved or if they're in regions that are relatively conserved in mammals, that means that these regions probably play an important biological role, right? And that's why they're conserved, because of natural or of negative selection, right? So that's another source of evidence. And then we have SNPs that have that there is evidence that they participate in biochemical processes in the cell, meaning that they probably regulate some genes or participate in the transcription process somehow. And in this figure, they stratified this set of SNPs into 3 levels of confidence: high, medium, and low. And you can see that the green blob intersects with all of these other annotations. So this gives you kind of an idea of how we can think about these functional annotations. And now I'm going to very briefly go over the types of functional annotations that I'm going to speak about for most of this talk. Not all of them, but very briefly. What are the types of functional annotations that we often think about? So first we have genomic region, whether a SNP is in a coding region or in a promoter or stuff like that. And chromatin state is a very important functional annotation because we know that SNPs in open chromatin regions are more likely to be involved in biological processes. And so we have information coming from DHS, DNase hypersensitive sites, or predictions based on histone marks. And we can also think about histone marks as an annotation in a very explicit way, so stuff like methylation or acetylation. So this is another source of evidence that this SNP plays a role in some biological process. And we have regions that are evolutionary conserved. And so these are probably important SNPs. and enhancers or super enhancers, these clusters of enhancers, which increase the likelihood of a transcription factor binding to a gene. And we have a few others, which I will mention later on, but these are the main ones that I'm going to speak about. And I should mention that this data, a lot of it comes from two very big consortia called ENCODE and RoadMap. So these are amazing, amazing consortia which collected lots of these functional annotations and measured them. And even though the papers are a few years old, most of the data that we use still come from these papers. All right, so these are our functional annotations. And now let's speak about what we can use these functional annotations for. And so I'm going to speak in this talk about 3.5 different tasks that we can do with these functional annotations. So the first is understanding genetic architecture, and I'll explain what this means later on. And a kind of a sub-goal of that is identifying disease-relevant tissues and cell types. So if there is some disease, we want to ask what are the most relevant cell types that drive this disease. So that's one task, or 1.5. The second task is identifying causal variants, which variants are causal for some disease or trait. And the third is genetic risk prediction, so trying to predict phenotype or trying to predict risk of some disease based on genotypes. And I'm going to show you how these functional annotations could help us perform all of these tasks. And before diving into the nitty-gritty details, I just want to mention that everything that we're going to speak about in this lecture is going to be based on polygenic models. So I already tried to convince you that almost all traits were polygenic. That's what you can see here in the bottom right, sorry. In fact, we get more and more evidence that everything is polygenic. So there is a very influential paper, perspective paper, that was published almost 3 years ago, saying that maybe we should not use the term polygenic, but use the term omnigenic. And this paper made a claim that almost every gene probably affects almost every trait. And that's because of gene-gene network. So there's kind of a butterfly effect where if there is a variant in one gene, so because of gene-gene networks, the effect will propagate to other genes and will eventually propagate to all of the directly causal genes, meaning that every gene is at least indirectly causal for each and every trait. And it's a controversial paper, but the bottom line is that we think that most traits are very polygenic. And so in this lecture, instead of looking only at these genome-wide significant SNPs, the SNPs that are above the red line here, for which we typically have at most hundreds of SNPs for a typical trait nowadays, we're going to simply look at all SNPs, millions of SNPs, and not even make the distinction whether we think a SNP is significant or not. And I go into more details later. And I want to mention two very important concepts for the rest of this talk. So the first concept is SNP heritability. SNP heritability is defined as the proportion of trait variants that can be causally explained by all SNPs in the genome. And this is an important quantity. This basically asks how genetic a trait is, right? How much is it influenced by genetics? Because we know that some traits like height are extremely genetic, and other traits like, for example, BMI or cholesterol levels are less genetic. So that's SNP heritability. And the second concept is functional enrichment of an annotation. So it's defined as follows. So it's a ratio. On the numerator, you have the proportion of trait variants that is explained by SNPs in some specific annotation, maybe, for example, by SNPs in coding regions. That's our annotation, maybe. And on the denominator, we ask what is the denominator, what is the proportion of SNPs that are in this annotation. So for example, for coding SNPs, we know that the denominator is 1%. I already told you that. And then the question is, what's the value on the numerator for some trait? If it's large, then that means that SNPs in coding regions are functionally enriched for that specific trait. And this gives us a clue into the underlying genetic architecture. If we can find for some trait what are the most important functional annotations, then we'll know maybe where to look and how to try to understand their underlying biology. And I'm going to show you some specific examples later on. All right, so that's, Yes, sure.
Because I think it's a good time to stop the recording and restart again. It's
Yeah,
a new place.
yeah,
Do people have questions?
sure.
Okay, I think it's very
Yeah,
clear.
I
I'm
do, I
gonna,
do have a
oh,
question.
okay.
Sorry.
Sure.
Can you go back to your last slide? Like,
Yeah.
I'm not sure, like, what do you mean by the denominator, like percent of SNPs that are in the annotation?
Among all SNPs that are in the genome. So if you look at all the SNPs in the genome, how many of these are in the specific annotation that we're looking at right now?
Oh, yeah, yeah. Oh,
Yeah,
okay.
so for coding SNPs, it's approximately 1%.
Okay, great. I'm gonna stop the recording.
All right, so we're back. And so that was the intro. And now I'm going to speak about the first of our 3 tasks, which is estimating functional enrichment. So first, I'm going to show how I think is convenient to think about our functional annotations. So I like to think about our data as a matrix. So let's say that we have this stretch of DNA. And so we have a bunch of annotations. And so we can think about each annotation as a binary vector. It's usually binary because for coding, every SNP is either coding or not coding, right? And the same holds for other annotations like intron, open chromatin, histone marks. And we have some other annotations like whether a SNP is a cis eQTL or not, and others. So you can think about it as a binary matrix. And I briefly mentioned that we also sometimes use non-binary annotations, but in this talk, we're going to think about mostly binary annotations. So we have this matrix, and now I'm going to speak a bit more in detail about these annotations. So I already mentioned some of the annotations. So we have stuff like genomic region, which I already mentioned, and chromatin state, and histone marks, and conserved regions, and enhancers. But we can take a pretty broad and liberal definition of annotation. We can take every set of SNPs that we think is interesting and has some interesting property, and call that an annotation if we'd like to. And so we can have an annotation, for example, for bins of minor allele frequency, something that we often do in our group. So SNPs that have a low minor allele frequency, it means that maybe there is some kind of negative selection acting on them, meaning that they might be important, right? So we can have a bin of all the SNPs with minor allele frequency between, I don't know, 5% and 7%. So that's one example that we often use in our group. We often use annotations related to LD, linkage disequilibrium. So some SNPs are in linkage disequilibrium in LD with more SNPs than others, and that might be biologically relevant. And some SNPs are in recombination hotspots. And so these are other types of annotations. And we can also take prior information from previous studies, like whether we already know that some SNP is a eQTL or not. So these are all examples of annotations that we can use and we do use in our group. And all right, so now I'm going to get into the details. So let's start by discussing how we often model genotype-phenotype relationships. And it's a pretty simple model. It's a linear model. So let's say that we have some trait Y. So we have a vector of 6 people in this case. So we have 6 people. Each person has some measurement for the trait, which could be, I don't know, height or blood pressure. And we assume that Y is given by X times beta, where X is a matrix of SNPs and beta is a vector of SNP effects, plus some vector called epsilon, which basically captures everything else that's non-genetic, environment, environmental variables, and noise, and everything else. And I made it very simplistic on purpose. But it doesn't get much more complex than that. And 2 things I want you to notice. One is that the vector beta is very long, and that's because we have millions of SNPs, because we're looking at all SNPs in the genome. So the vector beta is very long, and the matrix X is very wide. The second is that the matrix X is, you can see that it has only 3 colors, white, gray, and black, and that's because we usually encode the data as if every individual can have either 0 minor alleles or 1 or 2 minor alleles in each SNP. So each entry in X says how many minor alleles in some specific SNP an individual has. All right, so that's our model. It's a pretty simple model.
I have a question.
Sure.
So can you go back and explain what? So is each block one SNP, or is it one
Yes.
person, or is it one?
All right. Sorry. So yeah. So rows are individuals and columns are SNPs.
Okay, rows are individual. Okay, columns
Yeah.
are SNPs.
Should have
Okay.
that. Yeah.
And then the different colors are just different values. Or different.
Yeah. So I mean, can you see my cursor, my mouse cursor?
Yeah.
Yeah, sure. So this individual has individual number 2 has 0 minor alleles in SNP number 2.
Okay. All right. Thank you.
Right. So let's move on. So this is our model, and you might be thinking, why are we using a linear model? That doesn't make any sense. Biology isn't linear. It's much more complex than that. And you're absolutely right. But even though biology is probably not linear, we still use linear models. And the reason is that they fit the data as well as any other model that we try using. So you can try using very complex nonlinear models, and they will almost definitely fit the model at least not better than the linear model. And it boils down to dimensionality, basically. So without going into too many details, when we have a very, very high dimensional problems, every nonlinear function can be approximated almost perfectly with a linear function. And so basically, there is no reason for us to not use linear models because they're simple and they fit the data as well as any other model. People are often surprised by that, but that's the reality of these models. And so this is our model. And now I'm going to put it here on the bottom right. And first, I want to demonstrate that under some simple assumptions, heritability is given by the sum of the variances of the SNP effect of the betas. So what does that mean? So we're going to make a few assumptions, and we're going to use what we call a random effect model. So basically, that means that we treat these betas as random variables in a probabilistic model. And we're actually going to take an extreme approach and treat everything here as a random variable, also the x's and everything. And we're going to make a few simple assumptions. So the first is that beta I has 0 mean, meaning that every SNP is equally likely to increase or decrease the phenotype. The second is that its beta is uncorrelated with basically every other variable, every other beta and every other entry in X and every epsilon. Which could be debated, but it's probably true, at least to a first order approximation. And then we're going to make two simpler assumptions. So we're also going to treat the x's as random variables, and we're going to assume that they have a 0 mean and variance 1. But that's not really an assumption. You could think about it as a definition, because we can standardize these x's because we can see them, right? They're observed variables. So we just standardize them to enforce this constraint. And similarly, we assume that the variance of y is 1. But again, that's a kind of a definition because we can observe y. And the superscript j, by the way, is it denotes a specific individual. So we have individual j and SNP I. All right, and now let's go back to our main claim. The claim is that heritability under this set of assumptions is given by the sum of the variances of the betas. And how do we prove that? Here are the basic steps. So you could write it with a few more steps in the middle. So basically, we first write down the definition of y under our linear model. It's just a linear combination of x times beta plus epsilon. And then when we go from the first line to the second line, I actually skipped a few steps here for for brevity. But we first, because of our assumptions of uncorrelatedness, we can decompose the variance into a sum of variances. And then we take the definition of the variance of a product, and we use the fact that we have 0 means for beta and x to get these very simple expressions. And now we know that the variance of each xij is 1. That's assumption number 3. And we basically get this line here at the bottom. And then it's equal to 1 because of assumption number 4, right? So it's pretty simple. And the bottom line is that the proportion of the variance of y that is explained by SNPs is simply the sum of the variances of the betas, right? Because if this is equal to 1, then the sum of the variance of the betas is the proportion out of 1 that is explained by SNPs. And so this is important for the next slide. So if you haven't been following, just take believe me that the take-home message is that the heritability is given by the sum of the variances of these betas. All right, and now finally, we're going to define functional enrichment of an annotation. And so we're going to use the following definition, that the functional enrichment of some annotation, which we're going to call C, is given by this quantity. So we have a ratio of ratios here, right? And so let's look at these ratios. So on the left, we have the sum of the variances of the betas or SNPs that are in annotation C divided by the sum of all of the variances. So for example, annotation C could be the set of SNPs in coding regions. So we ask we count the variances of SNPs in coding regions divided by the sum of all the variances. That's the left ratio. And the ratio on the right is the size of the annotation C divided by how many SNPs we have in the genome. So basically, how many SNPs are in the genome. And it's very similar to the definition that I showed you before. So basically, we ask, if you think about what this means, we ask, are SNPs in our annotation, do they have large variances compared to the annotation size? Meaning, if they have large their effects have large variances, it means that they tend to affect the phenotype, right? Because their effects tend to be far away from zero, right? Meaning that every SNP is likely to either increase the phenotype or decrease the phenotype. So we ask whether they have large variances compared to the annotation size. So the right ratio is, again, it's 1%, for example, for coding SNPs. So this is the definition of functional enrichment. And it's derived based on what I showed in the previous slide. And we can also think about an equivalent definition that is sometimes easier to think about, that the functional enrichment is given by this ratio. And basically, the interpretation is what's written here at the bottom. So it's the average effect variance in annotation C, in our annotation, sorry, oops, divided by the average effect variance. So basically, we ask whether SNPs in our annotation tend to have large effects on average, meaning that their effects are far away from zero. If they do, that means that our annotation is functionally enriched.
So I have a question here. So
Yeah.
the SNP effect, do you assume that the major allele doesn't have effect and it's only the minor allele that you are estimating as having a functional effect on the trait?
Right, right. That's a great question. So I was actually kind of flying to you before. That's the answer. So I said that we encode SNPs as 0, 1, or 2, but we actually then standardize them. And that's what I showed in this assumption number 3. So they're no longer 0, 1, or 2. Now they're just 3 different numbers that have expectation 0. So that means that that means that basically we just assume that the minor allele, having 2 copies of the minor allele is going to maybe increase your phenotype, and that means that having 0 copies is going to decrease your phenotype because of this standardization.
Mm-hmm.
And that's just a convenience, because it means that we just, you know, there is some constant term that we absorb into this entire model.
Yeah, so in some sense, the beta is assigned per SNP locus, right?
The beta, it's per SNP, yes.
Right, per SNP you have an effect. And theoretically, if the number is big, it means that it has a big effect on the trait. But the variance of the beta is across all the SNPs then? Or is the effect of estimation, you're not so confident about the effect of some SNPs?
Right, exactly. So actually, this is a great question. So the betas are unobserved. So we're not actually, in this part of the talk at least, we're not even going to try to estimate these betas. We're just going, maybe I should answer your question after the next slide, because
Okay.
the next slide will explain what we're actually going to do. We're not going to try to estimate these betas. We're just going to estimate the, you know, the generative distribution from which these betas are sampled.
Okay.
So maybe we can go back to this question after this slide. So, all right, so now I defined functional enrichment, but I didn't tell you what we're going to do. And now I'm going to tell you what we're going to do. So how do we actually estimate functional enrichment? So we're going to use a method called stratified LD score regression, or SLDSC for short. And this method was first published in a paper by Hilary Finucane et al. In 2015, Nature Genetics, and I personally consider that a seminal paper. It really laid the groundwork for a lot of analyses of how functional annotations affect phenotypes in a polygenic way. So it's a paper that came out from our group, and so this method proposes to extend the model. So it's now we have another model, a model for the variance of the effect of a SNP, the variance of beta I, conditional on the annotations of SNP I. So every SNP has a bunch of annotation, whether it's in the coding region, whether it's in a promoter, and so on. And it proposes, again, this very simple linear model which iterates over annotations. And for every annotation, it's multiplied by the annotation value, AIC, by some quantity that we call tau C, which is the annotation coefficient. So AIC is usually 0 or 1, right? Because it's binary annotations mostly. And we have these taus. And all right, so what do these taus mean? A large positive tau means that for an annotation means that the annotation is probably an important annotation because it makes the the effects of SNP in that annotation have be far away from 0, right? Because they have a large variance, meaning that they're far away from 0. So coding SNPs are probably going to have a large positive tau. And a large negative tau means the opposite. It means that an annotation is probably not important. So SNPs in closed chromatin positions are probably going to have a negative tau, meaning that their effects are probably going to be close to 0. All right. What we want to do is actually not to estimate these betas, but just to estimate these taus. These taus inform us of the generative distribution of these betas. And how do we do that? We're going to use a method of moments estimator. And this is the idea. So for every SNP, we can estimate its chi-square statistic under very simple linear regression. So we simply regress the phenotype on that SNP, and then we get a chi-square statistic from the linear regression. And if you write down the math, so you can show that the chi-square statistic is the expectation of the chi-square statistic under our model assumptions is given by this quantity. And without going into too many details, the conclusion is that we can estimate these taus using simple multiple linear regression. Everything here is linear. We just need to, to fit this, to fit a multiple linear regression, and then we get estimates of these taus. So it's very simple. I mean, the data is complex. It's used data. It's a complex problem, but the solution is surprisingly simple. We just do this multiple linear regression. The only slight complications is that we need quantities that look like this. And we call these quantities annotation-specific LD scores. So basically what this means is that for every SNP I, we need to get an estimate of the sum of the squared correlation between the SNP and every other SNP K, weighted by the annotation value of SNP K for annotation C. And so again, we call these annotation-specific LD scores. But one very nice thing is that we can estimate these annotation-specific LD scores from an external data set and what we call an external LD reference panel. We don't actually need to get access to the GWAS data itself to estimate these annotation-specific LD scores because they're not specific to an individual. They just describe the distribution of the SNPs in the population. And this is a very important property. And why is that? Because it means that we can estimate functional enrichment using only privacy-preserving summary statistics, which is a really, really cool, cool property. Because genetic data is private, and researchers can't share genetic data. I mean, they're not allowed to, and it's illegal. And there's been a real renaissance in statistical genetics by the development of these privacy-preserving methods that can use only privacy-preserving summary statistics to do these very complex analyses. So a researcher needs to only release these chi-square statistics, which don't say anything about specific individuals. And then we can just take these chi-square statistics that somebody else published online and to compute these annotation-specific LD scores from external LD reference panel. And there is at least one panel called the 1000 Genomes panel that you can just download online right now. And you can do all of this by yourself without requiring access to the actual genotypes of individuals. And this is a huge deal. This really accelerated science, this entire field of summary statistics-based methods. And hopefully, after this slide, this answered your questions about how do we estimate the variances of these betas?
Can I ask a question about?
Sure.
Because I'm not sure if I understand this right, but I'm not sure why you're taking a negative value for the annotation coefficient when the annotation itself is not so important. Whether that going to cancel out some of the annotation importance of the other annotations, if you understand what I'm saying, because the variance should be taking consideration of like the how this beta is going to change among like a certain range. But if you're adding up the positive and negative values, those are they going to cancel each other out and give you false.
So that's exactly the goal. So if a SNP, I mean, we usually have positive taus. We have very few examples of annotation with negative taus. But I mean, I'm trying to think about a hypothetical example. Let's think about a simple example. If a SNP is both in a coding region and also in a conserved region, then it's probably going to have a larger effect than a SNP that is only in a coding region but not in a conserved region. So these annotations really add up. I mean, we have multiple sources of evidence that add up together to determine whether a SNP is probably going to have a large effect or not. So coding plus conserved is larger variance than coding but not conserved. And you can think about maybe, I'm trying to think about a hypothetical example. So maybe, I don't know, if a SNP is in a coding region but is also in a closed chromatin state, I'm not sure if that's possible. But anyway, if it is, then the closed chromatin state will kind of cancel out the fact that it's in a coding region. Does that answer your question?
Yes.
Right, cool. Yeah, so these annotations work together. I mean, we look at all of the annotations jointly, not only one at a time. That's a good question. All right, so I mean,
histone marks, do you take all of the histone marks as one track, or you do this one, for example, one cell type as one track? Because for example,
right.
conservation is only one, but K27 acetylation could be liver in brain.
Yeah. So this is a good question. These are actually two questions, I think. So the answer to the first question is we take multiple different types of histone marks, and they're often indeed correlated. So often we get very correlated annotations, but we still fit them all together. It's perfectly fine, doesn't bother us too much. It might increase the standard errors a little bit, but usually it's not a big problem for the type of things that we want to estimate, because it doesn't increase the standard error of the functional enrichments. Because the functional enrichment, we don't care about, if you have two competing annotations, you don't care about how you would divide the taus between them. So that's the answer to the first question. And the answer to the second question, whether we do this in a cell type specific manner. And the answer is sometimes. I'm actually going to show results in a few slides. But for the most part, the answer is no. For the most part, we take either annotations from blood, or we kind of take the union across many different cell types. And I mean, that's a decision. It's not always the best decision, but the reason is that we usually just don't know what the relevant cell types are for most traits. And I mean, usually it's not the main goal of our research, but we do sometimes carry out analyses where we take cell type specific annotations, and we're going to show an example in a few slides.
Okay, thanks.
All right. So now I'm done with showing you the method and the theory, and I'm going to show you some results from the original 2015 paper. So here again is a list of annotations just for your reference. I'm not going to actually go over them, but these are the main annotations that were studied in that 2017 paper. Maybe this also answers your first question. So you can see that we have multiple histone marks here, right? And all right. And in that paper, they analyzed 17 traits using only summary statistics. That's a big deal. So you can see we have traits like height and BMI, anthropometric traits, and some biochemical traits like LDL and HDL, and diseases like type 2 diabetes, schizophrenia. We have behavior traits like ever smoked and educational attainment. So it's very diverse. And you can see the sample sizes, they're tens of thousands, sometimes even over 100,000 people. So there was a lot of data. And all right, so let's look at some of the results. And I'm going to jump straight into the main punchline, actually. So this is, I think, the most interesting figure from that paper. So what you can see here is that they took 5 representative traits, that's in the legend here: height, BMI, agent Menardson, schizophrenia, and a meta-analysis of immune traits. And they took 5 representative annotations out of, I think, maybe 54 annotations. And you can just see the estimates of the functional enrichment. So the y-axis is functional enrichment. And so you can see that coding SNPs tend to be very enriched for heritability, which isn't surprising. What might be surprising, is that conserved SNPs are even more enriched for heritability. And for example, you can see that in BMI, SNPs in conserved regions are 20-fold enriched for heritability compared to the average SNP. So conserved regions play a major role in BMI, for example, in body mass index. And you can see that we got smaller enrichments for histone marks, though some of them are still statistically significant. We get depletion, the opposite of enrichment for SNPs in repressed regions. So almost half of the genome is in repressed regions of closed chromatin, and indeed we see that these SNPs have smaller effects on average than the average SNP. And maybe the most striking result is this huge red bar here. And what you can see here is that enhanced SNPs in enhancers are incredibly enriched for autoimmune traits. They were, in this case, 39-fold enriched compared to the average SNP. And this, I think, was a surprising result. I'm pretty sure this was a novel result. And that shows that enhancers play a very major role in immune traits. And this really gives us a clue about where in the biology we should be looking at when we study these immune traits, right?
Quick question, how do you define enhancers? Because usually K27 acetylation define enhancer. In this case, you have a different approach to define enhancers?
Yeah, that's a good question. And I'm going to be honest, I'm not really sure about all the technical details, but you can see that they're called Phantom 5 enhancers. So it's taken from a study called Phantom 5, I think. And I admit that I'm not really fully up to date about how they define these enhancers.
Yeah.
This was way before I joined the group, but you can find all the details online.
Thanks.
And yeah, I think that's the most striking result. And now I mentioned before cell type specific annotations. So here is an example. That's another figure from that paper where they did take cell type specific histone mark enrichments. And this is cool. This can actually help us identify what are the relevant tissues or cell types for some disease or trait. So they took these 4 histone marks that you can see here, and they looked at up to 118 cell types, and they grouped them into cell type groups, so 10 in total. And then they estimated whether a cell type is important for some trait by creating an annotation that consists of the cell type specific histone marks for that cell type. And then they fitted the model twice for each trait. Once when including this new annotation, and once when not including this new annotation. And then they asked whether the model fit was significantly better when they did include this new cell type specific annotation. And they got some really interesting results that you can see here. For example, you can see that for schizophrenia, they got a really significant result for central nervous system. So SNPs, with histone marks in cell type specific in this cell type specific annotation are really enriched for heritability. That's what it says basically. And for height, the most relevant cell type is connective and bone. And for rheumatoid arthritis, it's immune trait, sorry. I don't know if it's surprising or not, but it's stuff that people have been thinking about, that this is very direct evidence, coming directly from the data without any assumptions, that these are the most important cell types for these traits. And here are a few others, and there are a few others that I personally consider surprising. For example, central nervous system is very important for the trait ever smoked. So the trait ever smoked has a very strong genetic, behavioral component. And it's also very important for BMI, for body mass index. There are really interesting results here, in my view. And I also mentioned that there was a follow-up paper, kind of a follow-up paper in 2018 that also used data from cell type specific gene expression to try to hone down on the most important tissues and cell types for disease. And that's it for this part of the talk. So any questions?
It's interesting that genetically people are predicted to have X number of years of education, which is kind of sad to think about. The years of education is predicted by genetics.
Yeah, so it's very controversial, but when you look at the data, you can see that the fraction of variance of years of education that you can actually predict from genetics is less than 2%. So we should be careful about the interpretation. The fact that we find relative enrichment in some SNPs doesn't mean that the actual heritability is high. We just ask how much the heritability is divided across these specific SNPs, but it doesn't mean that the actual heritability is high. So it's not all bad. Yeah, but it's actually very controversial. There are papers about that. Yeah, but it is very heritable. It's, I don't remember, it's maybe close to 10% heritable or something like that.
Okay.
Yeah.
I'm going to stop this section recording.
All right. We spoke about estimating functional enrichment up until now, which is kind of a broad question of, you want to understand, genetic architecture, where heritability is coming from. Does it come from SNPs in enhancers, in coding regions, and how to partition heritability? And what I'm going to speak about now are two more specific tasks that we can do by leveraging functional annotations. And so the first of these tasks is identifying causal SNPs. And let me give you a brief background. So we know that GWASs by themselves identify associations, not actually causal relationships. So here is another Manhattan plot, like I showed you in the beginning. And Manhattan plots typically look like this. So again, the SNPs are arranged on the genome, and we get these peaks. So we get a peak here and here and here. And why do we get these peaks? It's because of LD. It's because probably we have only one or a few causal SNPs in this peak, but because all of these SNPs are correlated, so they all get a significant p-value. So what we actually want to get is not the associations, but want to estimate causality. So what we actually want to get is something that looks like this, where here on the y-axis, we don't have the p-value. We have what we call posterior causal probability, which is a common measure. And I'm going to also use the term PIP in this part of the talk. So PIP is posterior inclusion probability. And for some reason, this term stuck. So it's a probabilistic measure between 0 and 1, which asks how likely is a SNP to be causal. And you can see that as we moved from this figure to this one, the signals became much sparser, right? Its peak became much sparser, only one or a few SNPs survived. And so hopefully these are the causal SNPs, and these are the ones that actually drove the signal in these loci, and all the other SNPs, just tagged along because of LD. Not really causal. And that's what we want to do. We want to identify these causal SNPs. This is a very important problem. This is actually the real goal of GWAS, to identify the causal SNPs, because this gives us a clue into the, the biological mechanism, and then we can start thinking about treatments or drugs. Pharma companies are really interested in estimating these causal probabilities. And so that's what we're going to do and speak about in this part of the talk, estimating these causal probabilities. And I just say a few words about LD, which is going to play a major role in this part. So LD is, as I say, our friend and foe. So just a few words about why we have LD, because of recombination. So here's an example. Let's say that we have a mom and dad and a child. So the child has two chromosomes, one coming from the mom, and that's a mosaic of the mom's chromosome, and one coming from the dad. And that's the reason that we have LD, because SNPs that are close by tend to be inherited together from parent to child, and so across many generations. And so that means that SNPs that are close by tend to be correlated. If you know that I have an A in that SNP, then I'm going to have, for example, a T in a close by SNP. And what does that mean? What are the consequences of this recombination? So there are good news and bad news. So the bad news is that we have often very strong LD between nearby variants, meaning that it's very difficult to identify the causal variant, because if you have two very correlated variables, then it's very difficult to identify which one is causal and which one isn't, right? By definition, because they're correlated, they look similar. But the good news is that we essentially have no LD between distant variants because of the recombination, again, because the process diverges quickly. And that means that if we write down the LD matrix, the matrix of these correlations, then it's banded. We're going to get zeros when we get far away from the diagonal. Sorry. And this is good news. This is good news because it means that when we want to identify a causal SNP in a specific region, we only have to look at this specific region. We don't have to consider SNPs that are far away, because they're going to be uncorrelated. They're going to be completely irrelevant for our task of finding the causal SNPs. So there are also good news, but we do have strong LD within a locus. All right, so these were 2 minutes about LD. All right, so let's get back to our main task of finding these causal probabilities. So what we want to do is called fine mapping. That's the term. So the idea of fine mapping is that we take two things. We take GWAS results, and we take a matrix of LD, of these correlations between nearby SNPs, and we combine these two things together in a probabilistic model to estimate these causal probabilities that we care about, these PIPs. All right, so that's what we want to do. And one thing that has been known by several people, and we often find that, I should mention that most of the results I'm going to show now are based on my own paper under review right now. So we know that fine mapped SNPs are enriched for functional annotations. So here is a figure from our latest paper showing, so what we did here is we ran fine mapping on many traits from the UK Biobank, and then we partitioned the SNPs according to these PIPs, these posterior causal probabilities. So blue are very strong confidence, meaning that these SNPs are very likely to be causal, and orange is kind of intermediate confidence, and green is weak confidence of causality. And we partition them, and then I'm showing here the functional enrichment across 5 representative annotations for these 3 groups. And we get results that are kind of what we expect maybe. So non-synonymous SNPs are almost 40x enriched. Non-synonymous fine map SNPs are almost 40x enriched, meaning that there are 40-fold more non-synonymous SNPs in the PIP greater than 0.95 SNPs compared to the average SNP. And we get similar results for conserved, for SNPs and promoters, for histone marks, and we get depletion for repressed SNPs. All right, for repressed SNPs, right? So this gives us a clue that annotations could really guide the fine mapping process. So what we're going to do now is to do the opposite. Instead of first running fine mapping and then asking what's enriched, we're actually going to leverage these functional annotations to improve the fine mapping process. So the idea is that functional annotations can tease apart SNPs in strong LD. And to see that, let's look at this example where we have this thread of DNA and we have these 3 annotations, coding, infron, and conserved. So we can say, without knowing anything, that these 3 SNPs are probably the most relevant ones, right? Because we know that they're coding and they're conserved and they're not in an intron. So a priori, we can say that these SNPs are probably the most relevant ones. And so we want to leverage this information. And here is a cartoon example of how we're going to do that. Let's say that we have this Manhattan plot, and we know that probably one of these 3 SNPs here in the middle is causal, but we don't know which one. But now let's say that we know that this SNP here in the middle is coding and conserved, and the other 2 aren't. So this gives us evidence that this SNP is more likely to be causal than the other 2, right? So that's what we want to do. We want to leverage these functional annotations to improve the fine mapping process. And so I'm going to describe a method that we developed called Polyfun that performs fine mapping with polygenic functional priors. And it's not the first method to do that, but it improves over previous methods in scalability. So previous methods were limited in terms of scalability in one of 2 ways. Either they could only collect information from a few loci or from a few functional annotations. And Polyfun combines lots of recent developments to kind of aggregate information from all across the genome, from millions of SNPs, to weigh each SNP optimally according to its annotations, according to hundreds of annotations. And we're using hundreds of functional annotations. We could probably use thousands, though we didn't try that. And so let me give you a few more details. So we're going to take a Bayesian framework. So the idea is that we're going to take 2 things. We're going to take GWAS data. We're going to take these functional annotations data. And we're going to use these 2 things to estimate a prior distribution of beta I. So for example, SNPs in coding regions, their beta I is probably going to be non-zero, right? And then we're going to use the GWAS data again, together with the prior distribution of these betas, to get an estimate of the posterior distribution of beta I. So it's a completely Bayesian approach, and for every SNP, we're going to get a posterior distribution of beta I. So I should mention that in contrast to what I showed in the previous part of the talk, now we're actually going to try to estimate these betas directly, not just the generative distribution. And I'm speaking about these betas from that model here on the bottom right. All right, so this is just kind of, the scheme of what we're going to do. And I should mention that we're being careful not to use the same GWAS data twice. So we're putting part of the GWAS data aside for the posterior and another part for the prior. But it's kind of technical, and I'm not going to get into the details. So a few more words about Bayesian modeling of genotype-phenotype relationships. So here is our model again, and we're going to make an assumption that each beta I, each effect of each SNP, can be either 0, meaning it's a null SNP, or it can be normally distributed, meaning it's a causal SNP. And then we have two tasks that we can do. And the first is fine mapping, what I'm speaking about right now, which is trying to identify the non-zero beta I's. So maybe, for example, only these 3 SNPs with the circles are causal. Maybe they have non-zero beta I's. And the second task, which hopefully I get to speak about in the final part, is risk prediction, meaning that we want to predict the phenotype for an individual based on the genotype. And the common task is that we simply want to estimate the posterior distribution of beta I, and then we can use, for example, the posterior means to do these two tasks, right? So fine mapping in, under our model is asking what's the posterior probability that some beta I is non-zero. All right. Any questions? All right. So we're going to speak about fine mapping now. So let's speak about the model for one more slide, and then I will show you the results. So here is our model. I already wrote it in English, but now it's written in math. So for every SNP, we have its effect beta I, conditional on its annotations. And we assume that it could be, so the generative distribution could be either it's drawn from a normal distribution with some probability pi that depends on its annotations, or it's 0 otherwise. So we're going to assume kind of a sparse model. And basically our task is estimating pi, right? If we can estimate pi, then we estimated the prior causal probability of that SNP, which we need to use in our Bayesian model. And so in order to do that, we're going to relate the prior causal probability to the variance. And if you write down the math, it's pretty simple. You can show that pi is proportional to the conditional variance of beta I, conditional on annotations. And this is cool. This is something that we already know from the last part of the talk, right? We already have the SLDSC model, which decomposes this variance according to the annotations of the SNP. So now we're back in familiar territory, and we already know how to do that, right? So if we can estimate these taus, these annotation coefficients, then we can estimate this variance, and this will give us an estimate of the prior causal probability for that SNP. So we basically enrich the model from before by adding more assumptions. And so again, our problem is that we need to estimate the annotation coefficient tau c, and we already know how to do that, right? Almost, I just mentioned that we're not using the regular version of SLDSC. We're using a regularized and robustified extension of that method. And the reason in one sentence is that this regularization gives us more accurate estimates at the cost of some bias. So it really depends on what's your goal. But in our goal, we are willing to get some slightly estimate bias in order to get more accurate estimates. It's a regular bias variance trade-off. And so, all right, so this was a very brief rundown of how the method works. So briefly what we're going to do now, I'm going to show results from our fine mapping analyses, which took data from 49 traits from the UK Biobank, which I already mentioned. It's an amazing resource. So the average sample size was 318,000, kind of crazy. We looked at millions of SNPs with all SNPs with minor allele frequency above some pretty lax threshold. And we used 187 function annotations from a model that we call the Bayesian LF model that we often use in our group, that includes a pretty broad set of coding, conserved, regulatory, math, and LD related annotations. And we analyzed thousands of loci for each trait. And we used a method called SUSY to do the posterior inference, published in Barra Archive in 2018, a really, really good method. So the idea is that we focus not on the posterior inference by itself, but on getting very accurate prior estimates for each and every SNP, so that we can improve the fine mapping power. So the idea is that, again, if a SNP is non-synonymous, then it's a priori more likely to be causal, and hopefully that should help guide the the fine mapping process. And I'm going to show you now an example that should hopefully give you some intuition about how this works. All right, so this is just an example, a real example. So this is a fine mapping of height, and we have some locus on chromosome 2. So the x-axis is just the the position of the on this region, and for every SNP in this region, we have two shapes. we have a circle and a square. And I should mention that the y-axis shows the posterior causal probability. So every SNP basically has two posterior causal probabilities. One according to the model that uses the function notations, which we call polyphantasy, and that's the circle, and the square is the posterior causal probability without using these functional notations. And all right, and the color of each shape shows the prior causal probability that we estimated based on these functional notations. And so maybe you can already see that there is one SNP here which has PIP close to 1, meaning that it's very likely to be causal. And we could identify that SNP using our, our model, which uses the functional notations. But the model that does not use functional notations, it didn't have, enough confidence to say that this SNP is causal. It gave it posterior causal probability of around 0.35. And why is that? Because it didn't know that it's a non-synonymous SNP. So it's a non-synonymous SNP, and this turns out to be important information. I mean, it's also lots of other things, but in particular, it's non-synonymous, and this apparently was very useful information. And so this gives you an idea about what's going on. What's going on is that we use these function notations to help us tease apart SNPs in strong LD. Even though this SNP is in strong LD with the other ones, it has enough evidence because we know that it's non-synonymous, and maybe it's lots of other annotations. I just chose to write non-synonymous. All right, so this is the idea. Function notations can help us tease apart this strong LD. Here is another example. It's kind of similar. In this case, the phenotype is red blood cell count, and the top SNP was in a promoter. And again, this was enough evidence to make this SNP go all the way to the top. And the model that did not use function notations, even though it thought that this SNP is a good candidate, it wasn't confident enough about it. In fact, it wasn't even the top candidate SNP according to the, to this method, that the one with the squares. All right, so this is the idea. And so we ran this fine mapping method on lots of traits, and in total, we found 32% more fine map SNPs than non-functionally informed fine mapping. So using functional annotations turns out to be a pretty big deal in this case of fine mapping. And here you can see a breakdown of how many SNPs we found per trait. It's less important, but the bottom line is that we found around 3,000 SNPs. And what you can see here is a visualization of all the SNPs that we found on the genome. So every circle here is a SNP that was confidently fine mapped for at least one trait. And I guess the most interesting thing that you can see here is that whenever you see a bunch of circles next to each other, it means that the same SNP was fine mapped for multiple traits that are genetically uncorrelated, meaning that there is a technical definition, but it means that basically they have very different genetic architectures. And we found some SNPs that were really pleiotropic for lots of different traits. For example, this SNP was fine mapped for height, BMI, waist-hip ratio after adjusting for BMI, red blood cell count, total cholesterol, blood pressure, lung capacity, and balding. These are really distinct traits. I mean, they have very little to do with each other, and it was fine mapped for all of them. So this gives us insight into what maybe are, the really important SNPs that play a role in very fundamental biological processes. So there's lots of info that we can get from this fine mapping. And I think that for time constraints, I'm just going to finish this part of the talk and just skip the third part. I think otherwise it's going to be too rushed. Is that fine?
Actually, because it's recorded the video, I think we should just record everything, and students who has to go to another class can still go, but I would, at least for myself, would love to hear the rest of your talk.
Oh, all right, that's great.
Okay.
Fine with you. All right, sorry for that. Always difficult to manage the time. All right, so I briefly want to show you something else that we can do with our Bayesian framework of the fine mapping called polygenic localization. And this kind of ties together the fine mapping and what we did in the previous part of the talk, which is about partitioning heritability, asking how much heritability is coming from SNPs in coding regions and so on. And so we tried to bridge these two things together, and we came up with the concept of polygenic localization. And the idea is that we're going to quantify how much heritability is explained by the K SNPs with the strongest effect. So once we did this fine mapping, we actually not only did phi mapping, we actually estimated the posterior distribution of the effect of each and every SNP. And now we know which SNPs are the strongest, which ones are the second strongest, and so on. And this will give us a clue about how many SNPs explain a given proportion of heritability. And in this case, we're going to use the annotations indirectly because we already used them when we did the posterior inference, right? And so just a very few words about how we did this polygenic localization. One thing that we cannot do, we cannot just take the polyphony estimates. So naively, we can just take the estimates of beta I or beta I squared, the magnitude of beta I from what we already did, and use them to rank the SNPs. But we can do that because of a common statistical phenomenon called Wiener's curse. And Wiener's curse basically says that you should never rank items and then do statistical inference on the top ranked items using the same data, because these top ranked items are, some of them got lucky by accident, and that's why they're top ranked. And so your estimates are going to be biased. And so instead, it's kind of technical, we used a separate data set to be absolutely confident that we're unbiased. So what we did is we estimated this beta I squared, the magnitude of the effects of each and every SNP using our method polyphon. And then we partitioned SNPs into bins of similar beta I square. And then we re-estimated the heritability in each bin using our favorite method, SLDSC, but using different data. And in this case, we used basically all of the European individuals in the UK Biobank that we didn't use in the original phi mapping analysis. It's kind of technical, but anyway, we use different data. And one question that we thought would be interesting to do is to ask how many SNPs does it take to explain 50% of the heritability? So H squared G is heritability across different traits. And the answer is that it really varies widely across orders of magnitude. So you can see here that the range goes from less than 100 for hair color all the way to over 500,000 for chronotype. Chronotype is a property of being a morning person, whether you're tired in the morning. So it's a very polygenic trait. Unfortunately, you can't fix that. And yeah, there's a huge range here. And this really gives us an insight into which traits have a more complex biological architecture than others, or genetic architecture than others. And so this all fits together into our posterior inference framework, which gains a lot by using these functional annotations. And one other thing that we can do with this is we can actually look at individual traits and ask how much heritability accumulates across the genome as we add more and more SNPs. So what you can see here is I'm going to show 3 examples. So on the x-axis, we have the number of top-ranked common SNPs for, in this case, hair color. And on the y-axis, we have the proportion of common SNP heritability that is explained by these SNPs. And when you look at this, it looks like we need 0 SNPs to explain 50% of the heritability of hair color. But if we zoom in on where most of the action is, so we can see that it's not really 0. It takes 25 SNPs to explain 50% of the heritability of hair color. So for hair color, we got very strong polygenic localization. But hair color is kind of an extreme example. So if we look at, for example, height, which is a more representative trait, we can see that it takes 3,400 SNPs to explain 50% of the heritability, which I still think is a really interesting result. Height is considered a really polygenic trait. It's kind of a textbook example of a polygenic trait. But what we're showing here is that it takes only, way, way, way less than 0.1% of the genome to explain most of, the genetic signal of height. So we think that's a really interesting result. And again, we have a range of traits. For the trait for which we got the weakest polygenic localization is chronotype, or the property of being a morning person. And here you can see that according to our estimates, it takes more than 500,000 SNPs to explain 50% of the heritability. So there's really a huge range here. And all right, so that's it for the second part of this talk. So I'd be happy to take any questions.
I think for the time, maybe we will
right.
continue with the next section. I'm going to stop the recording and restart again.
All right.
Depends on whether we have time at the end. Is that okay?
Right. All right, hi again. We're now going into the third part of this talk, which is going to actually be much shorter. And now we're going to leverage functional annotations to improve genetic risk prediction. And yeah, I'm going to run a bit over time, so sorry for that, but hopefully only by a few minutes. Now we're switching gears and we're speaking about genetic risk prediction. And there is a concept called polygenic risk scores, which is basically the same thing. So polygenic risk scores is the process of taking somebody's genotypes and try to predict some future phenotype based on the genotypes. Conceptually, it's simple. People have been speaking about it for many years, but I think only in the last 2 years has it really made it into the prime time. And this is a very influential paper from the Broad Institute from 2018, showing that polygenic scores for common diseases are sometimes as effective as monogenic mutations. And specifically, they showed that they can use polygenic prediction methods to predict risk of cardiovascular disease, and then people with elevated risk can start taking statins, even though clinically they look fine. Maybe they should start taking statins. So this actually has lots of clinical relevance, and many people think that genetics will have a lot of clinical relevance in a few years because these polygenic risk score methods really are making it into the prime time and they're becoming very effective. Here is another influential paper from the same group about polygenic prediction of weight and obesity. Again, if we can predict that from an early stage, maybe we can do some kind of intervention. So these polygenic risk score methods really have lots of potential clinical utility, and this even made it into mainstream media, New York Times, and so on and so forth. So these are polygenic risk scores. But these polygenic risk scores also have a dark side, and the dark side is that they don't translate well right now across ancestries. And this was highlighted in a recent paper, again from the Broad Institute. So here is a figure from that paper. So this figure shows basically that our GWASs are predominantly focused on European ancestry individuals, much more than the relative proportion of Europeans in the population. So basically what we know is mostly stuff about European genotypes. And the second thing they showed is what they did here. So they built prediction models using European ancestry individuals, and then they tried applying these predictions on individuals of non-European ancestry. And what you can see here on the y-axis is the relative prediction accuracy compared to Europeans. So Europeans, by definition, got 100% of their prediction power. But as you go to other ancestries that are more and more genetically divergent from Europeans, the prediction accuracy drops. And this is a very concerning problem. And so now I'm going to show you how fine mapping and functional annotations can help us mitigate this problem. So here is a cartoon figure of one of the main reasons for this loss of accuracy. So let's say that we have two populations, maybe European and non-European. And let's say that we have two SNPs, SNP1 and SNP2, and some trait. And SNP2 is a causal SNP for this trait. But you can see here that SNP1 is in LD with SNP2 in this population, but not in this population. This is a very common phenomenon. LD patterns are population specific. So what's the problem? The problem is that SNP1 is predictive of the trait in population 1, but not in population 2, right? So if you try to build a prediction model using population 1, you might use SNP1, but then when you go to population 2, suddenly SNP1 doesn't have any predictive power, right? That's a problem. And so what can we do about it? So what we can do is to use functionally informed fine mapping, or I mean any fine mapping, but functionally informed is even better, to implicate SNP2 to figure out that SNP2 is actually the causal SNP and not SNP1. And then we can use SNP2 for prediction instead of SNP1, right? So if we knew that SNP2 is relevant, why would we ever use SNP1 anyway? So that's kind of the ideal goal. That's the utopia. What we're actually going to do is we're just going to go back to our Bayesian model, which you already know. And we're going to now, again, compute the posterior inference to get a posterior estimate of these betas. And then we can do risk prediction. So risk prediction means that we get the genotype of some individual, and then we can simply compute the weighted sum of xi times beta I. Xi are the SNPs of this new individual, times the posterior mean probably of these betas, and that's our prediction, right? So you already know all about the method. I don't have anything to show you. So basically, I'm almost going to make it on time. So we did that, and this is work in progress. I should mention that. But we basically took our posterior estimates, and we use them to perform risk prediction. And a few facts. So we used, again, the UK Biobank, so up to 337,000 people of British ancestry. And we then applied the predictions on individuals from the UK Biobank of non-British ancestry. And we did that so far for 15 genetically uncorrelated traits. And I'm just going to show the results. So you have 4 rows for 4 different populations. You can see here the sample sizes for each of these populations. And basically, you have 2 columns of results. The third is the relative accuracy compared to Europeans. So as you expect, the relative accuracy drops as the population becomes more and more genetically divergent from European ancestry or British ancestry individuals. But the interesting thing for the purposes of this talk is what we gain when we use functional notations to improve our posterior estimates of these betas. And you can see that the improvement increases as we become more and more genetically divergent from Europeans, because that means that it becomes more and more crucial to actually use the true causal SNPs. And so for African ancestry individuals, we got a 13% improvement when we used these functional notations compared to when we didn't. And yeah, the final part of this talk is really very short. So that's all I have to say about that. So any questions about this part before I just wrap it up in one more slide?
Question here is, do you use in the polyphen here, do you use a cell type specific enhancer score, or you just combine all the histone marks together as one?
Yeah, that's a good question. So we did not use setup specific annotations. We could, and maybe we will do that in the future, but we didn't do that so far. And again, maybe one of the main problems is that it's not clear what the relevant cell types are for each trait. And maybe that's a research question by itself. I mean, yeah, but I'm sure that, you know, there could be a research project which does that and improves the predictions even further.
Yeah,
but we just didn't do that.
Thanks.
All right, so just let me wrap it up quickly. So I hopefully convinced you in this talk that functional annotations are a good thing. So they can do lots of stuff. They can help us elucidate polygenic trait architecture by partitioning heritability. They can help us identify disease-relevant tissues and cell types. They can help us improve the power of fine mapping, and they can help us improve the accuracy of polygenic risk scores, which are the 4 parts of this talk, basically. And that's it. I'll be happy to take your questions.
Yeah, this is really great. Thank you so much. I'm going to stop the recording.